Hi, everybody, and welcome to part three of this week's lecture. And today, what we're going to be doing is we're going to be focusing on proteins. So, by the end of this week, what you should be able to do is you should be able to identify the roles of protein in our living organisms. You'll also be able to list the different classes of proteins. So, just like in carbohydrates, we knew that we had different types, our monosaccharides, disaccharides, polysaccharides. We're going to have something very similar to these. Again, we're going to have our monomer and our polymers. We're going to see exactly why the structure of proteins and the shape are going to dictate the function and how they're going to be linked together. All right, so before we go ahead and get started and look at all those things, what we're going to do first is we're going to look at proteins in the human body. Now, this right here, you can see I have a image that shows you all the different types of proteins that can be found within the human body. And one thing that's important is proteins are going to be incredibly abundant, so they're going to be everywhere, and they're incredibly diverse forms of macromolecules. Again, there are going to be tons of them everywhere, and they're going to be very diverse. So that means that not only do we have many different or amounts, but we have many different types. Some great examples are going to be things like these antibodies, which are going to be our proteins, and they're used to fight foreign invaders. We have actin and myosin, which are going to allow for muscle movements. We have cytoskeletons, network of protein filaments and tubules, keratin found in our hair and nails, collagen gives our skin strength, cell membranes, enzymes, which are going to be really important. We're going to look at these and see how they speed up reactions. We have hemoglobin, blood, which is going to transport oxygen. And again, we have many regulatory ones too, like hormones, which are going to be proteins that are going to elicit hormonal responses like insulin. So what I have for you is I took a lot of those things and I categorized it down for you. So you can see we have the different type of protein, so such as enzyme, transport, structural, hormone, defense, movement, and storage, and then examples of those, and then what those functions are actually going to do. So one of the things I would definitely do is definitely highlight, star, circle, or do something along the lines to make a note of this chart, because this chart is going to be definitely really helpful for you to categorize all of those different types of proteins and their functions. All right, so let's go ahead and take a look at protein, specifically synthesis and hydrolysis. So remember, just like in our carbohydrates, hydrolysis, remember, is the addition of water in order to break something down. And then when we're looking at synthesis, we're going to be taking away water to make something. Okay, so over here is an example of what that might look like. So let's say this is protein in your diet. So let's say you took a protein shake, you ate a piece of steak, chicken, anything along those lines, and you'll notice that we have a very long chain of these. So in this case, they're not in our monomer, they're in our polymer form. So we want to break these apart. So remember, we're going to use hydrolysis, so H2O, to break down those proteins into our monomer. And in this case, our monomer is going to be our amino acids. So now we have all of these individual amino acids, and then we're going to go through dehydration synthesis. Remember, dehydration, the absence of water, synthesis in order to make something. So now we can utilize all of those in order to synthesize and make whatever proteins we need in order to help our body function and work. So again, protein synthesis, we're going through the two similar processes that carbohydrates went through in order to break things down and build them up. Hydrolysis, adding water, we add that water to break this apart. And then dehydration synthesis, in order to make something, we're going to take all of our monomers, we're going to take away water, and then we're going to synthesize and produce something new. So let's take a look at our proteins and our amino acids now. So as you know, amino acids are going to be our monomers. They're going to be our smallest forms. Now, in humans, we have about 20 different essential amino acids, and they're going to be very important for many different things. They're going to build up thousands of proteins, which are going to be important for those human structures and our functions. Now, when we look at our amino acids, this is typically going to be our amino acid structure. You can see we have our hydrogen here, we have our amino group, we have our carboxyl group, and then we have an R group. Now, one thing that's important is this R group is going to dictate what type of amino acid or function it's going to be. So again, we have those 20 unique amino acids, and we do have our unique R groups, which are going to give our amino acids very specific chemical properties, and it's going to give it that specialized function. Now, I'm sure you're wondering, what are those 20 amino acids? So right here are going to be those 20 different amino acids. Again, this is something that is kind of important for you to remember as well. There is kind of an easy concept or easy way to memorize or remember these different type of amino acids. So you can see over here we have two categories. We have an abbreviation and we have the actual amino acid group. Now, these again are going to be the 20 important amino acids. We have valine, we have glutamine, we have histidine, all of these different things. But you can see that we have an abbreviation. The way the abbreviation works is it's typically the first three letters of the actual name. So you can see we have alanine here, and then ALA is going to be our abbreviation. Leucine, same thing, LEU, LEU here. So that's an easy way to kind of remember this. You're not going to necessarily need to memorize these, but you do need to be able to differentiate between the abbreviation and that amino acid. So again, to summarize this up, we have 20 different amino acids. They're all going to have a very specific kind of chemical structure to them, and that is going to be thanks to this R group over here. That R group is going to be unique, and it's going to give those amino acids those specialized or specific chemical properties. All right, so now that we went ahead and seen exactly what our amino acids are, proteins, and things like that, we can go ahead and see how our protein structure is going to begin to determine its function. So the way that this works is whenever we first start off building our proteins, we have to take a bunch of amino acids. Remember, our amino acids are monomers, meaning it's going to be our smallest form, or in this case, one single unit. Now, what's going to happen is once we take a bunch of these monomers and we start adding them together, you can see that they're going to begin to be chained all together. We're going to create something known as a polypeptide. Okay, so poly, remember, is going to be many, and peptide. What's happening here is our covalent bonds, which are bonding all these together, are going to be known as a peptide bond. So in this case, we're binding many together, many amino acids together in order to form this polypeptide. Now, as of right now, this is non-functional. And the reason why is because it doesn't have its specialized shape yet. So this is going to be known as our primary, or in this case, our first level. Now, it's not until we get into our second parts where we start to take these proteins and start folding them down. You can see over here in our secondary or our second part, our proteins are going to start becoming more shaped and specific to their job. And then as things move on, they're going to fold into each other more and more into step 3. And then finally, until they become our mature functional protein. In this case, it fully has a shape now, and it's able to produce or do what it's supposed to do. A really good way to kind of think about this, or a really good analogy, would be a key. So if I have a key right here, imagine the primary as an uncut key. There's no teeth yet, so it can't really do anything. And then as we're moving down further and further, think about it as it begins to get its shape more so, and then until finally it's got its complete shape and it's finally able to open whatever it needs to open. So that's just kind of an analogy in order to connect the idea of a non-functional polypeptide, many monomers together, and as it's getting its shape, it's going to become more useful. All right, so this is going to conclude the first portion of our protein lecture. What I want you to do is I want you to take a second and see if you can answer these questions, and then we'll kind of look at them together as a class, and then we'll move on to the second part of this lecture. All right, welcome back, and let's go ahead and check this out. So when it comes to this, some of the main ideas that we're trying to grasp today is our polypeptides, our amino acids, and all of the different monomers and polymers that we're trying to connect together in order to get the proteins. So looking at this here, you can see that, remember, we have our amino group, and then we have our carboxyl group. Now, whenever we're adding these together, we're linking them together, we're trying to produce a polypeptide. And in this case, when we're linking things together, we're adding covalent bonds to those amino acids in order to form a polypeptide. When it comes to this, remember we have those two different groups. We have our amino group and our carboxyl group. And remember, the R group itself is the thing that is going to make it specialize, that specific chemical structure. Name this type of chemical reaction when we're trying to separate things apart. I'm sorry, when we're trying to add things together. Dehydration, remember, is the absence of water, so H2O, and then synthesis is we're trying to create something. So this case, you can see we're trying to add these two together, and what we're going to do is we're going to go ahead and subtract out our water in order to bring it down here, which is going to be synthesizing or producing one big thing. So in this case, it would be dehydration synthesis, also known as anabolism, and adding or building something together. So it would be anabolism, and it would also be dehydration synthesis. Finally, in the addition to the peptide, what molecule is produced apart from this reaction? Remember, water, whenever we're going through dehydration synthesis, we are subtracting that water out. All right, so let's go ahead and check out examples of collagen structures. So another thing that's really important when it comes to proteins is collagen. Collagen is going to be incredibly important. It's going to be the most abundant protein in humans, making up about 1/3 of their body. Collagen is a protein that's going to be very strong, flexible, and fibrous proteins. And again, it can be found in many different things. It can be found in our skin, our internal organs, our muscles, eyes, our connective tissue, as well as our bones. So again, these are examples of structural proteins that are going to aid and help within our body. So again, proteins incredibly important, incredibly abundant, and incredibly versatile when it comes to the human body. So when it comes to collagen, the reason why that's going to be so kind of strong, flexible, just like in the past slide says, strong, flexible, fibrous, all of those things, is because it has to do to the actual structure of it. So looking at this over here, you can see that individual collagen polypeptides are kind of shaped like long strings. These collagen strings are weaved together to form fibrils. These fibrils are then weaved together to form strong fibers that are then weaved together even further to build our tendons, ligaments, and things like that. So collagen fibers are weaved together into cells called fibroblasts. So you can see here, these are primary amino acid chains. Remember, amino acid monomers added together in order to make our polymer. You can see that these are going to be 3 linked collagen chains together. So we're starting to weave this. So we have one strand. Now we have many strands. We're going to weave that even further into these bundles to make them even stronger. So again, this is a great example of collagen, which is our protein, showing structure and function, again, being incredibly versatile and being found in many different places within our body. So now we're going to go ahead and take a look at another example of proteins, which are going to be known as regulatory proteins, or in this case, hormones. Hormones are going to be really important. The reason why is because hormones are going to control body processes. So a great example is going to be insulin, which is going to control those glucose levels. Remember in our previous lecture, we talked about how the body is going to break down our carbohydrates into our monomers, and you can see that a lot of that was glucose. So again, insulin is going to help control the amount of glucose within the body. And the way that it's going to do that is it's going to utilize receptors on our target cells. So kind of what that means is you can see that we have hormone A here, and then we have receptor A down here. These are going to be our cells, and you can see that they're going to be completely different. The reason why is because, again, it's kind of like a lock and key, or in this case, just like a puzzle fitting together. Only hormone A is going to match with receptor A. Hormone B is only going to match with receptor B. So again, in order for this process to happen, the receptor, which is a protein, can only accept the hormone that matches it together. So again, just like a puzzle piece, only those certain pieces fit together because, again, the receptor is specifically shaped for those hormones. One really important thing and one really cool thing is by understanding these shapes and structures of these regulatory proteins, we're actually able to kind of manipulate or synthesize drugs in order to kind of alleviate certain things. So again, drug companies study these protein shapes in order to design drugs that can turn on and off these specific cellular processes to combat diseases. A great example is going to be when you take painkillers, or in this case, Advil or Tylenol. They're going to go ahead and they're going to fit into these receptors, kind of fooling it in order to turn on and off the sensation of pain. So in this case, again, hormone B is very specific and has a very specific shape to receptor B. In this case, for those drug companies, they might be synthesizing something else that looks something along the lines of this. So this part will still slot in over here, so it can turn on and off the cellular process. So this might not be exactly the same as hormone B, but it'll be close enough in order to do this. Moving on to example 3, we're going to look at enzymes as proteins. So enzymes are going to be really important, and we're going to be specifically looking at lactase as an example. So enzymes, such as lactase, are going to help speed up certain processes. So if you are not lactose intolerant, that means you have the enzyme lactase, meaning that you're able to break down milk sugar. Remember, milk sugar was our disaccharide, so it was made up of two pieces. So your enzyme, in this case, is able to take that, right? So this is the enzyme, it's able to take it in. It's able to, remember, subtract H2O because we're going through dehydration synthesis. And now you can see it's broken apart that bond and it's separated out those two pieces. So it's helping speed up reactions with those enzymes. Now, a really good way to think about this is it's kind of like a lock and key. This substrate is going to be very, very specific to this enzyme, specifically the activation site. They have to fit exactly together in order for the enzyme to do whatever it needs to do. So that means if we had a substrate that looked like this, a block, it would not be able to fit into the substrate because, again, the substrate is very specific to the activation site, and the activation site is very specific to the substrate. So again, it won't just take any type of substrate. It needs to be that specific substrate in order for that enzyme to happen. Now, this isn't just going to be a specific kind of case study where it's only happening with lactase. Living organisms contain thousands of enzymes, so your body is consistently and always doing this. And the cool thing about this is after it's done whatever it needs to do with the substrate, the enzyme doesn't go away, right? It doesn't disappear. It doesn't become inactive. It's ready to accept another substrate in order to continue that process. So substrate is essentially any substance that binds specifically to an enzyme. So an example is lactose with lactase. And then the product of that is just what's being produced. So again, this was our substrate, and this is what was being produced in our product. In this case, we're taking our lactose and we're breaking it apart into our two individual carbohydrates, or in this case, our monosaccharides. So again, products is just substances that are produced at the end of the reaction or process. In this case, we're taking our lactose and we're producing our galactose, our glucose and our galactose. All right, so now that we went ahead and talked about that, we're going to jump back a little bit specifically at the structure and the shape of proteins. And the reason why that's going to be important is because we're going to be looking at something known as protein denaturing. So as you know, proteins are going to have a very, very specific shape. Right here, it's going to be functional. The way that it's specifically folded is going to give it that function. Now, it can't go through a process known as denaturing. Denaturing essentially means that you're changing the shape. So again, proteins have a very, very specific shape they need to be in order to work. And whenever you apply certain external factors, such as pH, temperature, or salt concentrations, it can change the shape of this protein. And by changing the shape, you essentially are going to make it no longer functional. Because again, those shapes are very, very specific. So Denaturing, again, is the chemical interaction between, or the chemical interaction that is going to disrupt the shape that's going to make it non-functional. So an easy way to kind of see this is an egg. Whenever you first crack an egg, you'll see the yellow part in the middle, and you'll see the outer sides are going to be white. But once you start adding or increasing the heat, you're going to see that it's going to change shape and it's going to change its consistency. No longer is it going to be kind of jello-like. No longer is it going to be viscous. It's going to be nice and hard. It's going to change its shape. It's going to become white. And then the yolk in the middle is going to start turning yellow. Again, the reason why is because, again, our proteins have that specific shape. Once you start changing it, you're doing something known as denaturing it, and now it's no longer functional. So again, denature is a change in protein structure that leads to the change in function. Okay, and then our next thing we're going to look at is going to be mutations and protein functions. So as you know, mutations are going to be a change in something or a change in code that's going to cause an abnormality, which is going to cause a completely different protein to be produced. So what we're going to look at is the example of sickle cell anemia. Now, before we can talk about the mutation, we need to understand exactly what sickle cell anemia is. Now, this right here is a normal red blood cell. As you know, this is going to be protein. It's incredibly important to have the specific shape. You need to have this round shape in order to increase surface area. You need to have this little divot inside in order to maximize the amount of oxygen you can transfer. The oxygen transferring is going to be done by hemoglobin. Okay, so both of these are going to go hand in hand. Very important that this has this shape in order for it to transfer that oxygen. Now, this is going to be the normal sequence. We have LEU as one amino acid. We have THR as another amino acid. We have PRO, which is another amino acid. In normal blood, we have glutamic acid, GLU, and then we have another one, GLU. Now, when it comes to sickle cell anemia, there's going to be a complete different shape to the cell. You can see that it's a lot slimmer. It's got this sickle shape. And because of this shape, what's going to happen is it causes it to be very sticky, meaning that it's going to stick to each other. It's going to get caught in capillaries, things like that. And we have our mutated version. So this is what it's now looking like. It's clumping together. It's not the right shape. It's not going to hold very much oxygen. So it's going to cause a lot of health issues. And the reason why it's causing these health issues is because there's going to be a mutation. So unlike normal, where our glutamic acid here is supposed to be GLU, once we get down here, it's going to be a change in the amino acid, which is going to cause huge issues. So this is the mutated sequence. You can see that this is still the same, LEU, THR, PRO, but when it gets down to here, we have an incorrect amino acid. So instead of GLU, now we have VAL, which is causing that change. So because we have this small change in our amino acid sequence, it's no longer giving us the red blood cells we want, but it's giving us our sickle cells. And because we have that sickle shape, again, it's going to be changing the function drastically, causing lots of health issues. It can cause blood clots. It can cause people to be very short of breath. It could cause life expectancy to decrease, things like that. All right, so that is going to conclude our lecture for proteins. What I want you to do now is I want you to go ahead and take an opportunity to test your knowledge. There's going to be a couple of these, so I'm only going to go over specific ones. There's going to be one on the second slide. I'm going to talk about the structure and function, just to kind of drive home the idea of how important that's going to be. So I won't go over all of these. I'll just go over some of those pieces. Okay, so we're going to go ahead and get started. I'll give everybody a couple seconds to pause this, and then we'll check out the answers. Okay, so these are going to be the answers for the first round. The first thing I kind of want to just touch on is, remember, our monomer is our smallest group. So remember, it's just our singular unit. So in this case, it's going to be our amino acids, because triglycerides are going to be for our fats. Monosaccharides, we know, are carbs. And then nucleic acids are going to be for DNA. So our amino acids would be for our proteins. The other thing I want to touch about is that R group, remember that R group with our amino and our carboxyl group producing amino acids is going to give us our distinctive properties. So this is what's going to give us our unique 20 amino acids. So that part is going to be true. This is going to be the second part of test your knowledge. So same thing, couple seconds to give it a try, and then we'll check out the answers. All right, so these are the answers for this. The one I really want to touch on is going to be number 2. So essentially what this question is asking is it's trying to research. So we have a biotechnic company that's researching a drug for diabetes. And what it's trying to do is we can see that this is going to be our insulin acceptor. And what's essentially happening is they have 4 different types of drugs that they're looking at in order to turn on and off this insulin receptor. So the way you would answer this is you're going to look at our specific shape, because remember, this is going to, our receptors are made of proteins, and these receptors are very specific to the hormones. So in this case, we need to find something that will be accepted within this section. So you can see we have that kind of moon curve shape. So we know that drug A can't because it's got that flat line at the bottom. We know drug B can't because it's pointed down. We know drug D can't because it has all these jagged edges. So in this case, drug C would be able to work because this would be able to slot right in order to turn on and off those reactions. So the main idea from this question is our receptors are very specific to their hormones. And then finally, this is our last slide. So same thing, there's only about 2 questions, so I'll give it a couple seconds, or 1 question, so I'll give it a couple seconds for you to pause it and give it a try. All right, and in this case, a monosaccharide is to a polysaccharide, right? Because you take a bunch of monosaccharides, because mono is 1, and then saccharide is sweet, and poly is many, saccharide sweet. So in this case, you're taking a bunch of these monosaccharides to produce a polysaccharide. It's the same for amino acids. We take a bunch of amino acids, right, to produce a polypeptide, which is going to be our protein. All right, and that concludes our lecture for proteins. I look forward to seeing everybody in our last lecture. So I'll see you in part 4.